Hello and welcome to the next video in the A-Level Biology series. In this video, we will discuss genetic engineering. In this video, we will cover the main principles of genetic engineering. What is recombinant DNA? The roles of vectors, restriction endonucleases, and ligases in genetic engineering. Genetic engineering is a technique used in science to deliberately modify a specific characteristic or characteristics of an organism using recombinant DNA technology. The main applications of which are producing large amounts of desired proteins, for example, insulin and human growth hormone, vaccinations, and to produce large amounts of antibodies for diagnostics and therapeutics. Recombinant DNA is when specialist enzymes are used to cut and paste together desired DNA sequences. This produces a sequence of recombined DNA. The basic principles of genetic engineering are: identify and remove a gene with a desired characteristic from an organism. Transfer the gene using a vector into another organism. The organism expresses the desired characteristic. This is known as a genetically modified organism, or GMO. The main steps of genetic engineering. First, identify desired gene, such as a protein coding gene. Isolate the desired gene from the chromosomes using enzymes called restriction endonucleases. Multiplication of the gene by polymerase chain reaction. Then transfer genetic material into bacteria for cloning using a vector, such as plasmids, viruses, or liposomes. Grow transformed bacteria on selective media to build up the stock of the desired plasmid containing the gene. Use marker genes incorporated into the new gene to identify cells which express the desired gene. Now to cover these important enzymes in genetic engineering. Restriction endonucleases or restriction enzymes are proteins produced by bacteria which cut DNA at highly specific regions. Restriction enzymes precisely cleave DNA into fragments at specific sequences of base pairs known as restriction sites. The original function of restriction enzymes is to restrict the infection of bacterial cells by viruses such as bacteriophages by degrading viral DNA and not the bacterium's own DNA. EcoR1 is a restriction enzyme which recognizes and cuts at the base sequence GAATTC. Sticky ends cut in an uneven way across the sugar phosphate backbone of the DNA, leaving an overhang of a base, which makes it easier for genes to recombine due to hydrogen bonding with the base overhang. Blunt ends are cut straight across the DNA with no overhangs. The same restriction enzymes can then be used to cut the plasmid vector isolated from the bacterial cells in order to create a plasmid backbone vector for our desired DNA to be inserted. This is important when annealing the cut desired DNA into the plasmid vector, which we will discuss later. DNA ligase is an enzyme used to splice or insert the desired gene into a vector. It catalyzes the formation of phosphodiester bonds in the sugar phosphate backbone of DNA, creating a recombinant DNA plasmid. As mentioned earlier, vectors are the devices scientists use to carry new genetic material into foreign cells. Some examples of vectors are plasmids. These are the most common vectors for genetic engineering. They are small circular DNA found in bacterial cells separate from the chromosome. They are effective because they replicate independently. Plasmids transfer DNA into new cells in a process called transformation. Plasmids can be modified or naturally contain important markers for detecting transformed cells, such as green fluorescent protein, GFP gene, and antibiotic resistance genes. Another type of vector are viruses. Viral vectors have been used since the 1970s. They make very effective vectors due to having naturally efficient methods to infect cells, transferring their genome, which can then be replicated within host cells. Viral vectors are used in gene therapy, in which functional genes or proteins can be introduced to patients with diseases resulting from lacking functional proteins or genes. Viral vectors are specially modified to ensure they do not cause disease in the patient. The DNA within the virus is protected by the capsid, which is the protein shell of the virus. Some examples are adenovirus and adeno-associated viruses, or AAVs. These carry small DNA packages to host cells. The viral DNA is non-integrating. This means the DNA will not be copied in the production of daughter cells or be passed on to offspring. The DNA will persist for a long time in the host cells, which is good for when the viral vector can be injected into the patient, concentrating only on non-dividing cells, such as cells of the eyes, liver, and nervous system. Lentivirus and retroviruses are another example. These viruses contain RNA, but this can be converted into DNA within the host cells. The DNA will be integrated into the genome and therefore can be used as a treatment in dividing cells such as T cells and stem cells before being returned to the patient. Another type of vector are liposomes. These are less common methods of gene delivery. Liposomes are spherical vesicles made up of one or more phospholipid bilayers, which closely resembles the structure of cell membranes. Liposomes act as an envelope carrying material into the host cell where it can integrate with the cell membrane and deposit its contents inside the cell. They can be modified to contain DNA, mRNA, and drugs for delivery into target cells. We will now focus on the features of plasmids for use in engineering. Bacteria can take up DNA in a process known as transformation. Transformation occurs after the restriction and ligation of target gene to produce the new genetically engineered plasmid. Plasmids replicate independently of the main genome, meaning lots of copies can be produced. Plasmids contain restriction sites such as multiple cloning site where DNA can be inserted into the plasmid. The plasmid contains a promoter gene. The promoter contains a binding site for enzyme RNA polymerase, which catalyzes the production of mRNA in transcription of the new gene. Plasmids contain selectable markers such as antibiotic resistance. This trait is used to determine whether transformation of bacterial cells has been successful. If bacterial cells grow on media containing the antibiotic, this means they possess the resistance conferred from the plasmid. Now let's put all of these ingredients together to understand the process of genetic engineering. Genomic DNA is extracted from the target cell and the DNA plasmid is extracted from the bacterial cell. Restriction enzymes are used to cut at specific base sequences in the genomic DNA for the target gene and the plasmid DNA at the restriction sites. The target gene is inserted into the DNA vector or plasmid by ligation. This creates a recombinant plasmid. This plasmid is used to transform competent bacterial cells. Bacterial cells divide rapidly, creating large number of bacterial colonies containing the plasmid. Now to summarize. Recombinant DNA is DNA which has been constructed artificially and contains special sequences or genes from other organisms. The main enzymes used in genetic engineering are restriction enzymes, which are used to cut DNA at specific sequences, and ligases, which paste fragments of DNA together. Vectors are devices used to carry new genetic material into foreign cells. There are 3 types: plasmids from bacteria, viruses such as adenovirus, and liposomes. The main steps in genetic engineering are to isolate desired gene and extract plasmid from bacterial cells. Use restriction enzymes to cut the desired gene out of the DNA sequence and use the same enzymes on the plasmid to create the gene fragment and the vector. Insert the gene into the vector and ligate. Transform competent bacterial cells with the recombinant plasmid. Allow the cells to grow and divide, creating more cells with the recombinant plasmid. These can be induced to express proteins which can be purified later. This concludes today's video of part 1 of genetic engineering. Thank you for watching. We hope to see you next week for the final part of genetic engineering, focusing on polymerase chain reaction, gel electrophoresis, and applications in medicine and agriculture.